biology. And currently, I'm a principal investigator of the ministry grant, which is focused on cardiometabolic diseases and microbiome and metabolome of western lowland gorillas, especially those housed in zoos, it means in captivity. And we are currently in the last year of the grant. The grant was for 4 years. And this grant is a kind of continuation of my long-term collaboration with Dr. Andres Gomez from Minnesota University. Andres is currently, hopefully, in Minnesota University, at the Minnesota University. However, I have my colleague Davison here, and we established a new collaboration with, and I will rather just say it in Czech because I forgot the English name, Česká zemědělská univerzity, with Docent Havlík and his PhD, and I hope to say your name properly, Anna Asselani Bergo.
Exactly.
Yeah, she's here to present the Metabolone part. So we are going to speak about about the microbiome and metabolome and the cardiometabolic diseases in gorillas, which is a kind of proxy for the similar situation in humans. But you will know more after this talk. So I'm handing the word to my colleagues.
So nice to meet you. And as previously mentioned by Clara, we are going to introduce our work about the cardiometabolic disease in gorillas. So one of the major causes of mortality in the U.S. Zoo house gorilla is related with cardiac health. So the main cause is called fibrotic cardiomyopathy. So it consists that the heart, the muscle of the heart, gets fibrous, and the movement of the heart, it's not going to pump properly, and thus the gorilla dies because of this disease. And knowing, our question is why are they dying? How can we help them? And one of the, reading the literature in the for humans, there is a very strong, lately there, a lot of research is going on about the gut microbiome. And we know that it's highly influenced by the diet. So diet shape the gut microbiome. But the gut microbiome has also influenced a lot of physiological functions, such as it can influence the gut and how there is the permeability, the metabolism, but also it can be related with some inflammatory status of the liver, and it's connected with the brain, with the muscle, with the metabolism. But what is also interesting for us is it's also connected with heart cardiovascular diseases. So knowing this, our hypothesis is that microbiome and metabolome reflect the heart condition of the host. And this is what we investigated. And it sounds a lot of work, but this is where we started actually our project. So we collected fecal samples from all over the world, from gorillas. And here you can see, the collection. We collect them, we store them in ethanol, and we ship them in the different labs. And later, once we decided to follow 3 approaches to investigate how the gut microbiome could be linked with the cardiometabolic disease in gorillas. So one is the gut microbiome profiling. We did this with 16S sequencing, but also using the technique of the metagenomic shotgun. I will explain a bit later more about these 2 words. But also the metagenomic shotgun give us the possibility to know the pathways that were, the abundance of the pathways. Another aspect we wanted to investigate is the fecal metabolome profiling. So it's like a feces composition of the gorillas. I mentioned microbiomics, and it's used to characterize and quantify microbial taxa responsible for different structure, function, dynamics of the microbial community. Usually this is done in two different ways, using a 16S based approach, which actually consists in using one gene, more specifically a region of the gene. We use like V4 region. There is a sequencing, and based on, and then there is like the databases, and we can assign based on the sequence which kind of microorganisms are present. Similar concept is also for the shotgun metagenomic approach, but the difference is that this, before it was targeting one gene, but here it's using the whole genome. And as well, in this case, the sequences are then linked to the microbial community, and then we have quantification and characterization of this microbial taxa. Another word I have been mentioning is like metabolomics, and this is me like it aimed to understanding the network interaction between metabolites. What we did, we had our fecal sample, we did an extraction in water, and we placed the extract in a big magnet. The technique we use is called nuclear miniature resonance spectroscopy, which is recording the energy transition at nuclear level of the different protein in the molecules. Once, like, it sounds a lot, but actually it's ending up with a list of metabolites, which these metabolites can be classified like amino acid, can be sugars, or can be short chain fatty acids, like aromatic compound in general. As I mentioned before, we collected fecal sample for all over the world. So here you can see the map. So we have 48 adult gorilla from 11 different US zoos. Here you can see the contribution for all the zoos. And 37 of them was had a diagnosis of cardiovascular disease, and 21 of them not. 17 gorilla were from European zoo, like from 4 different European zoos. And also we had 20 adults of wild gorilla, which from 3 different habituated groups, where we collected the sample, two fecal sample in the dry season and 2 in the wet season. I didn't mention before, for the zoo house gorilla, we collected 4 different samples, 1 every week for 1 month. Now, I give the word to Sam to tell us.
So as you guys know, this was an international process, right, involving 3 continents, North America, Europe, and Africa itself. We wanted to see how these different systems, the microbiome and metabolome, changes based on geographics, right? So on the left here, we look at the microbiome. Remember those sequences that we used. We used singular gene with the V4 region of the 16S RNA gene, and shotgun metagenomics to sequence the entire genome. We took those sequences and applied those sequences to a taxonomic database to match up what species each genome matches to. So what that creates to us is a microbiome community for each sample we collect, because each microbiome has multiple taxa in it. And we can use those taxa to find the similarities and dissimilarities between and within cohorts or groups, right? So over here, we have a bunch of scatter plot points, right? But each point represents a microbiome of a single sample. And what we can see here is basically this is a dissimilarity matrix. The more closer 2 points are, the more similar those microbiomes are. The farther apart they are, the less similar they are, right? We also mentioned how at the title it says weighted. This is simply us looking at diversity. We're not just looking at what kind of taxa are in these microbiomes. We're looking at how much of the population is composed of each of these microbiomes, right? We're getting the demographic data of these microbiomes we're collecting, right? Like a census. You guys know what a census is, right? Yeah. So, what we found here with our data, statistically, of course, geographics impact the microbiome significantly for numerous reasons, right? Climate, topography, even the diets, right? Gorillas in the wild do not eat the same foods that are found in zoo house gorillas, right? Makes a lot of sense. The microbiome reflects that, with showing clear grouping of the seasonal wild groups on the right here, while the more zoo house individuals are all more clustered together. But remember, our project is looking at the similarities between the affected with CMD, cardiometabolic disease, and the unaffected, right? And although there seems to be a high overlap, when we perform a statistical analysis called permutational multivariate analysis, which basically looks at all the variables of data we know and sees how much each variable affects the microbiomes we see, we found that although that slice, a significant contribution, statistically significant contribution of the differences that we see within these microbiomes is based on the heart health of the individual. It was only 1%, but it was significant. And this is only looking at 16S data, so we're only looking at one gene to identify our taxa. The same things can be said about metabolites as well, though, right? But what's more interesting here is when we break down the metabolites and what kind of metabolites we find. We find that the wild type gorillas usually have more fermentation in their gut systems than the zoo house gorillas. And what I mean by fermentation is their gut microbiome and gut metabolome contains more ethanol, simple sugars that have been broken down into simple carbon, short-chain fatty acids, if you guys have heard those terms before. What this is saying, basically, is in the wild, the gut of the gorilla is a fermentation tank. It's constantly breaking down complex carbon structures like cellulose or strong, rich, sturdy stock of roots, and breaking it down into easily to absorb nutritional product. Well, on the other hand, we have zoo house individuals. They're creating alanine, methionine, butyrate. These are proteolytic fermentation. So instead of breaking down sugars, right, they're breaking down proteins. And those proteins are getting absorbed into the host. Does that make sense? One side, wild individuals ferment sugars more, zoo house individuals ferment proteins more. So we have an interesting story here. What this is telling us is that the microbiomes are not just changing based on being zoo housed or wild, they're changing based on really geography, like Europe and in the United States, and even health status, although slightly. And how that microbiome is changing is impacting what is being fermented. The microbiome is changing the function of the gut, and that function affects the host's absorption of product. But going forward, right, we're able to say that, but that's easier to show you what are those changes, right? I'm claiming that there's changes occurring, but I want to actually now show you what those changes are, right? We then used our shotgun metagenomic data, right, to do a more in-depth version of our analysis. 16S is only looking at one gene, so it's limited. We can't necessarily get an accurate read on every single taxa. So using whole genome sequencing does a much better job of that, because you're using the whole genome, right? It's a whole fingerprint, not just one piece of fingerprint, right? And what we found is this. First off, ASVs rarefied is basically amplicon sequence variances rarefied. All you need to know, ignore that value, that's just our paperwork. All you need to know is that's the number of taxa we found. And we found that there were significantly higher amount of taxa in CMD-affected individuals than unaffected individuals. But then, we wanted, again, look at how the population is composed. We want to look at how much of a certain taxa is taking up the population and the diversity of the microbiome. Right? And that's where the Simpson's index goes. The Simpson's index is a Simpson's diversity index. It's looking at the diversity of the microbiome. And what we found here was that now unaffected have higher diversity. That's weird, right? What that's telling us is that although there are more taxa within affected individuals, it's less balanced. There's only a few of those taxa that are taking most of the population. So you could say like 3 are taking 50% of the population versus in the unaffected individuals where those 3 are taking up only like 10% of the population. Right? The population is more evenly distributed between the different taxa within the unaffected individuals. Right? So the unaffected individuals have a more balanced microbiome. But now, let's see who are the main players here, right? Who's causing these shifts? And so what we did another statistical analysis and some other complicated stuff to find out the top 20 indicators of affected and unaffected individuals. These 20 taxa are the taxa that are clear indicators of differentiating CMV affected and unaffected gorillas, right? And we can say that this unclassified Protoviridea is mostly found in unaffected individuals, highly found more in individuals. This is a unique taxa that's mostly found only in individuals that are not affected by CMV. I then wanted to see how the diversity plots look at a more easier way to consume method. I took these 20 taxa, and I wanted to see their abundances out of 100, but this is at a percentage, so 0.25 equals to 25%, right? 0.5 is 50%. And I want to see how they change between all cohorts. And we can see here, right, Phocacola coprophilus, for example, right, it is in favor of unaffected individuals, and it's higher in unaffected individuals, significantly so. Is everything starting to connect now? You're starting to see a pattern here. The microbiome is changing based on the health status of the individual. And these are just the top 20. There's probably many more. So now that I've established to you that the microbiome is changing, and what in the microbiome is changing, we have to figure out what functions are changing. And we can do that using our shotgun whole genome sequences. Because those whole genomes have multiple genes in them. Right? Just like you, your whole genome has your eye color, your height, your sex, whether you're male or female, all that stuff is in your genome. We can do the same thing with taxa by identifying what their functions are, what their pathways are, by using their whole genome. So we did the same processes that we did with our taxa, but instead we're using pathways. Right? Other than this typo, we can find that in unaffected individuals, there are more pathways, meaning that in unaffected gorillas in the United States, there is more microbiome activity than in unaffected individuals than there is in CMV-affected individuals. And again, the same thing we said about diversity. The diversity in the pathways of the microbiome are more balanced. Than in unaffected individuals than affected CMV individuals. Right. And again, I wanted to show you just these 5 major indicators of really functions. Right. And these functions are fascinating because a lot of them, if you read these long names, I'm not going to go through all of them, are in the process of making butyrate, lysine, methionine. Look them up yourself on a website called Metapsych. Metapsych is the world's database for proteins and pathways. If you look up these pathways, that's what they do. We then took honest data using metabolites, and we also did a similar process of finding out what metabolites are really associated with the affected and the unaffected individuals. And all this volcano plot is saying that is if you cross this x-axis right here, you're more towards the right. You're more towards unaffected individuals. Right? You're found more often in unaffected individuals. While on the right, you're found in affected individuals. Right? So what this data is saying is butyrate, lysine, and methionine, and all these other proteolytic fermenting metabolites are found in unaffected individuals. Meanwhile, in affected individuals, we have saccharolytic fermentation. The behavior of the microbiome is changing more similar to wild gorillas when they start induced cardiometabolic disease. But then, we wanted to associate these data sets. I mentioned that these pathways create these metabolites. I wanted to see if a machine learning algorithm could figure out themselves. So we use a machine learning algorithm called block partial least square discrimination analysis, which is really complicated. That all it's doing is it's taking our data sets and overlapping them and seeing which variables that we have are associated with each other. So again, just like the plots I showed you at the beginning, right, with the scatter plots, 2 dots closer are more associated with each other. The farther apart they are, the less associated with each other. Make sense? And we can see here, again, a trend. On the right, you have unaffected individuals. You have all our pathways that are producing lysine, methionine, and butyrate. And on the left here, we have all the other metabolites and these taxa. Now what's really fascinating then is to see what taxa are associated with the pathways and the metabolites. Because that means, very possibly, these taxa, that's their main functions, right? This Prevotella coprophilus taxa might have a really high association with the process of creating methionine and lysine and butyrate. But here's where it gets really cool. In human research, right, we've done cardiovascular disease. It's one of the #1 leading causes of death, at least in America. Trust me, we're fat and eat too much sugar. Oh, our food's too good to deny it. But what we found is lysine, plasma methionine, and butyrates are preventers of calcification of your arteries, or vascular calcification. And what this means is, in vascular calcification, calcium builds up in your arteries. It makes it harder to pump blood. And one of the leading causes for cardiovascular disease and heart attack. Because your heart can't pump blood when your arteries are clogged, right? So these butyrate, lysine, and methionine prevent that, though, by helping the absorption of calcium from your bloodstream into where they need to go, like your bone marrow or your muscles. These, known in humans, is to prevent and keep people healthy. And they're found in unaffected individuals. In affected individuals, CMD, they don't have these blockers. So it's possible that within zoo-housed individuals, these bacteria are protecting these unaffected individuals from developing CMD. Or over time, the host metabolism changes, and they need to rely on these bacteria more. The point is, the microbiome is directly impacting the host's abilities, quality of life, and safety within captive individuals. So in conclusion, right, these preventive metabolites are found higher in unaffected individuals, right? They're preventing and practically protecting these unaffected individuals from developing cardiometabolic diseases and cardiovascular disease. The pathways associated with these metabolites are also found in a strong association with these metabolites. So it's not just random occurrence that we're finding these results, right? Machine learning algorithms have proven that these associations are real. The models have potential. Now, this model isn't perfect, right? We have only a limited amount of gorillas, and we only had so much time to work on this stuff. Well, this is the beginning of an idea that you can use the microbiome and metabolome as a predictor method to diagnose your zoo-housed individuals, to help treat them and care for them faster. Right? Because a gorilla workup is expensive. You have to use anesthesiology. You have to get a gorilla specialist vet. You have to get that vet to your your zoo. You have to do a lot of expensive work in time that can also stress and strain the animal's health itself. That's very dangerous and costly for zoos. But doing this, if you can just collect a fecal sample and you can help pick out individuals that are at risk that do not have these possible bacteria that protect them, you can select and make better choices in how you care for your gorillas. And this can expand to human care as well, right? Because if we can do this with primates, we can do it with humans, or any animal, or mammal even. So I would safely say the microbiomics and metabolomics as a tool to predict the health status of unaffected or affected individuals is very critical and a great success. I want to thank everyone around the globe for helping us, from Dr. Gomez for hiring me on this job, for Yaroslav and his lab team, including Anna, for helping get the metabolite data, Clara for helping us get connected with Africa so we can have wild sample groups, and all our trackers for helping us find these wild individuals. We could not have done this project without any of these people involved. Everyone was important. I want to also thank all the zoos that helped us, right? Because doing all this data while they're taking care of their animals during COVID was hell for them. So I want to really make you guys know that I appreciate, and we should appreciate what these zoos did. Because they had to care for these animals under a pandemic and make sure and get us the data we needed. That's all. Thank you.
So I'd like to thank Sam and Anna for presenting our project so nicely. And like as Sam said, that it's very expensive and very dangerous actually, like to anesthetize gorilla, because the gorilla may not wake up. And that is why, for example, European zoos are not anesthetizing gorillas for a kind of health checkups in comparison to US zoos who are doing this. So that is why we couldn't have the information about the cardiometabolic diseases in European gorillas. Because in Europe, they do anesthetize gorilla only if the gorilla has a problem. Yeah. So, any questions?
Yeah. Any questions for either of us, really? We kind of threw a lot of that at you. Yeah, what's up?
Thank you for this really interesting talk. So I have just a main question, if I understood it right. Is the microbiome change basically caused due to dietary changes? Or is it, or what's actually causing what? Is the heart problems causing the metabolomic and microbiome changes, or is it the opposite? Or what is actually the cause then of the microbiome changes?
So that can be a lot of things, right? You're right, the diet can have a great impact on microbiome, right? But also just age and the endocrinology, right? The kind of chemicals that are released from the brain can also impact the microbiome. It's really complicated. And like I said, our data is limited. We aren't able to look at that. So, but we want to use this as a springboard to say, like, future researchers, find out really what's going on here. Is the heart affecting the microbiome, or is the microbiome affecting the heart? Right? Either way, it's possible you can use the microbiome and metabolome to use it as a predictor. Right? That's what we mainly want to say. You can use the microbiome and metabolome to predict the health status of individuals. Right? But that's a great question. Maybe you can answer it in the future.
Yeah, and I would like just to add, it's so in Central African Republic there are people who follow a hunter-gatherer lifestyle, and this is considered to be a kind of like ancestral or original lifestyle we all live, and they have a different diet indeed, and these people do not suffer from cardiometabolic diseases. And with our previous and hopefully future research was also done like on comparison of the microbiome and metabolome of those people who are like following this traditional lifestyle and like and having a different diet, and Westerners. And then we had the people who were living partially in the environment, but they were Westerners. And like we think, well, I personally think like the key is the diet. However, it's probably not only the diet; it's also the environment, like the people live in. Because, for example, those Westerners who were living with the Bayaka people were not eating exactly but like probably like there was kind of like impact of the environment. Yeah. So you cannot, and those like those zoo housed or captive gorillas is something like us Westerners, and those wild gorillas are like, and I'm so sorry, like I don't like about this comparison, but I think it's important from many points of view. And those like Bayaka people, those hunters-gatherers, are like the gorillas who are following like the normal or the wild lifestyle. Yeah. So I think that, like it's diet, it's environment, and it's a very complex picture. Yeah, very complex thing.
Thank you. Any other questions?
So I was curious one more time, if I understood, you have the heart disease results only from the American gorillas, right? Or
Yeah.
like United States gorillas?
Yeah, because anesthesiology is a dangerous thing. Even
Yeah,
with
okay.
humans, right? That's why there needs to be an anesthesiologist, a person that specializes in a surgery room, not with the surgeon as well, right? Because if they give you the wrong dosage by even, I believe, like even a milliliter, you can die in some cases. Like that's it. You're dead. Oh yeah. So I mean, you can see, and considering that gorillas are threatened species, and we really want to take care of them, it's important to like not mess that up. So in Europe, they don't really do that often. In America, I guess we don't really care that much. Hey, we're careful. Hey, good. But yeah, so that's why we don't have the European data. We only have it for US based because we're the ones that diagnose it.
But the idea behind was that we work on the US zoos. We have affected and unaffected gorillas, and then we have these European zoos, and then we can like try to see if we can pick up those gorillas which are in the risk of these cardiometabolic diseases.
Correct.
Based on the comparison of the affected and unaffected gorillas in the zoos in America.
Anybody else? Person.
Thank you for a very interesting talk. I would like to ask, because microbiome health was a big issue, like it was the hot topic in like 1 and 2 or 2 years back. So are there any teams who are doing similar comparisons on humans? And are there any suspects for other diseases that this could be solved or more looked into about microbiome health?
Yeah, so I mean, microbiome wasn't a hot thing 2 years. It wasn't 2 years ago. It was hot in like the cultural zeitgeist, like socially, like you started hearing about like probiotics and healthy microbiomes in cultures and Facebook and Instagram or whatever, right? But in science and biology, it's everywhere. Everyone's starting to introduce microbiome into all their papers just because they feel like it's a requirement now. Yes, there are plenty of researchers. I don't know their names by heart. I apologize. I should maybe. That are researching many ways of how the gut affects the liver, how the gut affects the brain, how the gut affects the your oral cavity, how it affects even your eyes and including your heart, right? There was a paper published, I believe, about 3 years ago now, that linked the exact roots of how the microbiome changing affected your heart, in which case they found that when your microbiome produces a certain chemical, that's not one of these metabolites, it gets absorbed by the host, and that goes to the bloodstream, and then what it does is actually blocks calcium channels within the heart. And so it makes it harder for the heart to actually pump because the calcium channels are blocked. And so that raises the risk of cardiovascular disease because your heart's now having that work harder and with less effort. So again, calcium. It's always calcium that's causing heart issues, isn't it? So, but yeah, that's it. Anybody else?
Have you considered taking samples from gorillas in captivity from Central Africa that would be fed on the similar diet as Western, as gorillas from Western countries in the zoos?
Do I consider that? I consider it after I collected the data, yes.
Because
no, I mean, it's good. No, that's a great question. Yes.
That
I mean,
would dispute any doubts about a geographic location and the
gorillas which are living in sanctuaries in Africa, and like we've done some research on them regarding parasites and symbiotic ciliates, and they are like their diet is in certain aspects even worse than the diet of the gorillas which are housed in European zoos due to financial problems. Yeah, so they cannot mimic the diet they are eating in the wild. So they are feeding them mostly with a lot of starch, and you know, like the problem is it that, for example, if you feed them with a fruit, so the cultivated fruit has a completely different like nutrition values than the fruit from the wild, and regarding the like content of tannins and like phenolics, and also regarding the content of fiber. Yeah, and so they are normally like they don't live like, you must imagine it's like some kind of bigger zoo, yeah, with and they cannot like do it like semi-wild, so it's quite complicated. So I mean, like most probably, I think we would come up with a similar, I don't want to say even worse, similar results as from the zoos.
Does anybody have any questions about just generally what the microbiome is, or metabolome is, or what NMR is, or just what 16S or shotgun sequencing is? Does anybody have questions about those? Because we had to go over those quickly, I apologize.
We learned it already.
Oh, good. Awesome. That saves me time. Fantastic.
Yeah.
All right.
I had a question, not related to that, and probably not necessarily like related to the research so much. But with like the effective gorillas, what is like what's the output, or the like? Okay, you find out these ones have like the predisposition. Like, what is then done in ways of treatment or monitoring to protect them or to help them, or like treatment-wise, saying, okay, they have the disease, or okay, we predict they'll have the disease. What is then the next steps to that for the like gorilla itself?
Yeah. So in modern time, at least in the United States, we usually, after diagnosis is CMD, they put them on beta blockers. Like they give them treatments that are similar to like what you give a human that has cardiovascular disease or CMDs, right? Beta blockers, tell them to try to make them exercise more, eat the right foods, right? Those the same kind of methods for humans, right? For this though, it's mostly just about like being able to like save time and be faster at it. Because if you can catch it sooner, you can make sure the animal is not suffering as much.
You can put them on the treatment sooner.
Yeah.
Or you can see like, okay, the microbiome and the metabolome is indicating that this gorilla may have a cardiometabolic disease. So you can decide which animal you would anesthetize to do a more detailed and direct observation.
Right. That's it. It's just for quick here. I'm sorry, you got two already. That's yours.
So,
Yeah.
is there any relationship with their microbiome and the behavior which can be like due to the heart diseases?
Yeah, behavior, that's a good question. We don't have any behavior data, but that would actually have been really fascinating. But sadly, I think that would have made the zookeepers go insane. Because, I mean, obviously, as a gorilla ages, they're going to get slower, right? And they're going to be less active. And obviously, less activity, it leads to like less fitness. And less fitness, as we know in humans, is a higher risk of cardiovascular disease, right? So it's very possible. I mean, it could just be a case of like them being less active makes them develop heart disease, and that changes the microbiome. That's a completely valid possibility.
Thank you.
So I think good point. Good future research. We can just throw it in future research. Anybody else? I'm gonna pick you soon. So is there anybody else before I pick him again? By the way, I don't mean to like kick you out. You're doing great.
Could you maybe go to one of these slides with the charts? I was just curious. I actually wanted to connect with his question about the age. Can you go a little bit? Yeah, this is, or one more actually. Yeah, one before. Yeah, perfect.
Yeah.
This one. I mean, okay. I see, for example, in the Simpson index, like the how to say, the diversity is in an unaffected much greater. But let's say, I mean, do you have as well some other data? As you said, you don't have probably the age and stuff like that. Any other data which which could maybe tell us something different? Because I see here points are, I think, 4 or 5 of them which are really jumping out like some outliers or something which could maybe if you remove them, the significance could be almost even between the affected and unaffected in diversity or something like that.
Yeah, so we actually do have their ages. Right? We do have ages, and we do have the name of the individual as well, right? And again, we put that stuff into Permanent. Permanent, you can put as many variables as you want into it, and it'll spit out all those variables and how much it thinks each variable is contributing. Right? It did not, for at least the microbiome data, it did not find a significant contribution of age, which is actually pretty cool. And not sexy either. There's a past paper that said that it was a male-oriented kind of disease for creating cardiometabolic diseases. Another paper. The sample size was small though, but with this sample size, we did not see that the sex had any impact. So this is exclusively affected and unaffected issue. But good point, good question. Way to catch up. You'd be a good reviewer. I saw that. Did you have a question? Behind? Did you have a question? No? Okay, fine. All right.
Well, we are already on this slide. I have one more question about this change in the amount probably of these bacteria. Is it based on the number of reads which you found in every sample, or
relative abundance? It's relative. It's based on relative abundance.
Relative abundance of reads, right?
The relative abundance of the reads. So calculating the relative abundance with the microbiome normalizes things, right? Because some samples can have more DNA from it than other samples, right? And so we can have this really uneven reading. But taking the relative abundance of each taxa within each sample normalizes all the samples together. So we took the relative abundance of them and then calculated it out. So everything has this equal. So yeah. Any other questions? Okay.
So you assume that these free living populations, they don't suffer from these cardiometabolic diseases, or is it possible that they had the same problems and just the sick animals died already and you don't see them in the population because the selection pressure is very high? Or so can you see in this microbiome data some indications that some of these wild animals, they can, there's no
So you're asking basically how often do wild gorillas die of cardiometabolic disease?
Yeah, is it known or no?
It's not known really.
No, there has been I know that I've been searching, so the western lowland gorillas are not so well veterinary observed as the mountain gorillas, but I think I've seen at least one report, like from the mountain gorillas when they indicated some cardiometabolic problems. Yeah, but like if you consider how many necropsies have been done, like with the mountain gorillas and how well they are observed, it's quite rare. Yeah, so I think it would be good to anesthetize like a bunch of wild gorillas. But yeah, I mean I think we can't do this. And when we do an anesthesia I think there have been only very, very few anesthesia in the western lowland gorillas in the wild, and usually you have the rest of the group around you and you are happy to survive. Yeah. That you are not being killed by other members. Yeah, so it's
Better to work on rodents.
Yeah. Humans are much better.
Yeah.
It's rodents.
Yeah, no, I mean it always it always ends up with with all the people who are interested in that said but it's easier to work with the laboratory mice because you can kill them, you can observe them, but with the gorillas and with the wild apes, it's super complicated. But on the other hand, they are our closest relatives, and that's the huge advantage.
It's an evolutionary call. Just seeing how much does cardiovascular disease in us reflect to animals? What's the benchmark, right? So, hand first.
So when I looked at this figure, so it seems that the diversity or the variability of these few European samples is much higher than all American samples. Is it some artifact or is it something behind it?
I think it could be many things, right? There's factors of like zookeepers and how well they keep the samples. It's topography, right? I don't think Madrid has the same kind of climate as Prague. I don't believe so. I don't know. I've never been to Madrid. But like even like things can change the microbiome so much. Like even like the type of bird that lands inside the pen that they live in, right, can change that. It can change the plant life that's around, and the kind of foods and the dirt that the food grows in can affect the microbiome. All these little things, right? And so it makes sense that individuals from different nations have much more radical microbiome changes than individuals from the same country.
Maybe I can just add one more interesting thing. Is look at the metabolites that present in the fecal sample. The zoo from Prague and Madrid, they are like actually overlapping, right? You can see here that they are really overlapping the wild samples.
Yeah. So
This is quite interesting, but we don't really have answer about this.
So the metabolites from one zoo are unique, have unique concentrations from the levels of another zoo in Europe. Right?
Sorry?
So Madrid has unique concentrations of metabolites versus Prague. Prague has its own.
No, like Prague and Madrid, they have more similar stool composition to the wild.
Oh.
I mean, we can see overlapping of the samples.
I think it would be good if we had a more wild animals.
Yeah.
Yeah, but Betan, she did a research on a microbiome of wild gorillas, and and it was not so like dramatically different as we did expect.
Yeah. All right. Now.
Oh, is there anything with like comparable findings to yours in like model lab animals? Like,
Yeah. So I mean, most microbiome stuff started with lab mice, right? Basically, we first found these kind of trends in humans, and we're like, we should test it. And then they tested on lab mice, and yeah, we have a ton of this stuff like the location of the cage in the room changes the microbiome.
but like specific with like heart disease.
Oh,
and
yeah.
just, like,
Yeah, the lysine stuff that I mentioned was first found in mice. Like lysine was found as a preventer of heart disease in mice first. Yeah, so it's pretty cool. It's biology rule. But yeah, that's it. Really. How much time do we have left?
I mean, I think does anybody have a question?
Yeah. Does anybody want to talk about weird stuff? I don't know. You can ask a Yankee questions, like what I eat for breakfast, or if I like to play baseball.
Okay.
No.
I mean, it does. Okay.
Last one.
Yeah.
Is there any age? There is like heart disease with age of the gorillas, right?
Yeah.
Younger age or older age or.
So again, yeah, we ran Permanova, and Permanova didn't find significance there. Yeah. So it was not able to say like, oh, this variable age has significant contribution to the microbiome changes. So, yeah.
And Betan?
is this just a thing in like captive gorillas, or do you get it in like captive chimps or bonobos?
It's mostly a zoo-housed gorilla.
But there has been at least it's funny, during the course of our project there has been 2 captive chimpanzees in Czech zoos who died because of heart disease.
Yeah.
So and I would say it's in both gorillas and chimpanzees, but the gorilla programs are more organized.
Okay.
Yeah. So this might be a possibility. And and I know that I think I don't know if both of those chimps were on the treatments because they knew that they have a heart problem.
Okay.
and they were giving them the treatment, but still both died. One was a bit oldish and one was quite young. It was surprising.
Okay. And there's like no genetic link either, or it's not.
That's way out of the scope of our paper. You have to do your own future research on that. That would involve host snipping, right? Looking at the genome of the host and finding out the single nucleotide polymorphs that.
Yeah, but I mean, just even if there's links like with the heart disease being.
You mean in humans?
Or the gorillas.
It is found in humans.
I think it's found in humans.
It is found in humans, so it's probably found in gorillas too. We just, I don't know, we don't have gorilla genomes.
Wait, I mean, like there's going to be.
There's going to be.
There's going to be a study on whole genome. Well, there are some studies on whole genome sequencing in gorillas.
Yeah.
Yeah, but actually, like our team is participating with sample contribution to another huge study on whole genome sequencing in wild, but like it'll be wild.
Yeah. So, did you guys like the talk? Did you guys thought it was fascinating at least? I hope you guys liked it. Oh, that's worth it to me. Good enough.
Okay. Thank you.
Thank you, everybody.